It's videoing. Okay. Oh, I'm gonna get this like queued up too. Do you ever like when you're typing like your two hands get out of sync and so like whatever comes out is like totally garbled? That happens to me like all the time. Like one hand is going faster than the other, and it's like then all the words that normally should be like this get all like this, and it's all the letters, not all the words. All the letters that should be like this, like left hand, right hand, left hand, right hand, left hand. It goes. Okay. Anyway, so that's the thing that happens. Okay, so we left off last time talking about how sequences are derived. I spent the entire class accidentally talking about that, and I did not quite mean to. And then the next thing that we had to talk about then was what do we get out of that? What is the result of that? And the result of that is these sequences. Really just text formats, text files of sequences of genetic sequences. Usually if we're talking about genetic sequence A's, C's, T's, and G's, if we're talking about proteins, you have the 21 layer codes of the amino acids, and we'll have those. And yeah, considering how long I went over talking about genetic sequencing, I probably won't be talking about protein sequencing. Maybe sometime, but not right now. So that's what we get out. We also have this thing called the raw chromatogram. So this is generally from Sanger sequencing. Let me show you really quickly what that looks like. Some of my actual ones. Here we go. I'm going to open. Let's do this. Just open a random one here. That's a horrible one. So normally they don't look that bad. This is really bad. And so what we have here is all the peaks that you can barely see, but you can't really see them. That's probably good because they're a total mess. And this is one that I really actually couldn't do anything with. And in Eugene, what we're using, it has all that. It has the base calls up here and down here. And then the other thing it has for each base call, so each base call is what we determined that position's base to be. So just a base call. And in Eugene, what you have is you have an estimate of the quality or the confidence we have in that particular base call in this thing right up above it. So if you look at this one, they are all like really low quality. Go look at another one that I think is better. Hold on. Let's do this one. Oh, no, that's the sequence. Didn't want the sequence. The AB1. And all these are certain .AB1 files. There we go. You can zoom into there a little bit more. See, this is what you like to see. Nice segregated peaks. Actually, let me boost those up a little bit so you can see them a little better. So nice individual segregated peak, nearly nicely defined, not these big blurred peaks that just kind of like take over entire things. And then we look at the confidence of the, up here at the top, and it's all like really nice and high. That's what you like to see. Sometimes you actually get to work with that. Sometimes, a lot of times you don't. Yeah, so that's kind of nice. Anyhow. So that's the chromatogram. So that's one thing that you get, and those are usually certain .ab1 files, and those are really nice to have because then you just get, when you get the sequences in a text file, you have no idea just from that file most of the time. There are exceptions to this that we'll talk about. Most of the time you don't really have any idea about, okay, so what did the actual data, the raw data look like as they were sequencing this? What did that gel or the gel capillary look like? And the AB1 files give you an idea of that, and that way you can go back and look and see how confident you personally are in these, because in the end you are the one that has to defend it. Okay, and then sequences, the actual sequences themselves then are generally stored as a text file, and here's a couple of the most common formats. I'm going to run through these. We have FASTA format, FASTQ, which is really just a modification of FASTA format that includes quality information, not the actual peaks, but just like that bar information. We have nexus files and phylip files. There's actually a handful of other files, but these are the most commonly used ones. And most phylogenetic programs that work with phylogenetic stuff will work with one of these, at least. So let's briefly go through and talk about each of these, because you need to be somewhat familiar with these if you're going to work with them. It often happens that these files are a little bit wonky when they come from places, and a lot of programs are very specific about how they come out and how you read them in, and so there's not all that uncommon that you have to tweak them to make them work right. But we'll get to that, and that's something we'll talk about a little bit more in lab probably. Okay, so FASTA format, we can use this either for genetic sequences or protein sequences. It's used for both. And it follows a very standardized format here. Each sequence begins with a header that is offset by a is less than sign. So that begins it out. And you'll probably recognize this because it's a FASTA format that I gave you for the regular expression exercise. That was a FASTA format sequence. And then the sequence then follows whatever happens after that line that has the is less than beginning. And anytime you have an enter in there, it's ignored. So no one, the programs that read this don't really care about the enters. All those line returns are going to be removed. And so you could have the entire sequence on one line. Sometimes they are truncated. The very common way is to truncate them at 70 characters on each line, so you actually get something that's readable on most formats. And the formats that generally you'll see, or format extensions. Okay, so here's a crazy thing. These are all text files. And format extensions, for whatever reason, never really got formalized really well, and so you'll see all sorts of weird things as file name extensions. And it really doesn't matter what you use. You can use anything, and as long as you tell the program this is a FASTA format, it doesn't care about it. But commonly it'll be .fasta, .fa. I've also seen .fst, .fs, I think. Anyways, so let's take a look actually at that. I believe this is okay, so this is FASTA format right here. So again, you have at the beginning, you have is less than, and then a whole header's worth up here. So a couple different things in here, and each bit of information, not literally a bit, every section of information or type of information is then generally when you get this from GenBank, is going to be offset by a vertical bar. And so you have GI, vertical bar, a number, vertical bar, GB. Okay, so this one right here is probably the first one that you really need to worry about. GB is the database in which this was originally stored. So GB stands for GenBank. You'll also see EMB. You'll also see DDJ, a few others. The third one, and now this is where, no, the fourth one, becomes really important. This is what's called the accession number. This is in whatever database it's stored. They have kind of standardized formats for identifying a number that identifies that one specific sequence. That's called the accession number. And this is now the identifying number that if you ever use this sequence again, or if other people use this, they need to provide the accession number so other people can go back and find that specific sequence. So whenever you, for instance, use genetic data in a paper and you publish it, the typical practice is to go on and then deposit that genetic data onto one of these genetic repositories like GenBank, and that sequence will be assigned an accession number. You then report that accession number in your paper so that other people can go and get access to that specific sequence. And then also, oftentimes, you're not just using your own sequences, you want to compare them to other sequences, generally ones that other people have produced already. You go and grab ones from GenBank, and you also report what accession numbers of sequences you were using. And this is just kind of the nice, handy, straightforward way to do that. And then after that, we have kind of the specific, more human-readable information about this. So this is Octopus tetricus. isolate NZ1 12S ribosomal RNA gene partial sequenced mitochondria. It's just something that identifies it, gives you a more regular description that you can identify it. And this part, after this final section from GenBank, it's very dull. There's no standard for how it goes. Generally, but not always. Generally, the species name goes first, and then somewhere in here you will have the gene name after that, and then there's other information that is just all over the board. There's no real rhyme or reason to it. Okay. Any questions about FASTA? Excuse me. Okay, then we have FASTQ. FASTQ, so this is similar to FASTA, but it gives quality data is added. Now, this is the file format that's often used for high throughput sequencing because, as you're going to see when we go into lab, you're going to be evaluating Sanger data, which is generally low throughput and is fairly easy for you just to sit down and look at and evaluate how good that data is. However, that being said, if you're going through high throughput data, you now have, like, after one run, you have sometimes billions of individual base calls, and you are not going to sit down and read through those all by yourself. And so what happens here is the actual text file then has this quality data added to it so that you can now sort or pair your data according to this quality. And what these do is these are set up very similar to FASTA files, except instead of beginning with an @ sign, you have is less than sign, you have an @ sign that begins the header name. And then you have one line, generally one line for all the genetic data. So this is not, it's best practice not to include returns in the genetic sequence. And then you have a plus, and then down below there we have characters representing quality data. Now, the characters representing quality data are like normal characters. These are like alpha, like you have all the alphanumeric characters, you have all the punctuation marks, and you have other random marks. And these are, by just convention, these are mapped to different qualities. Oh, I should have grabbed a FASTQ file. I actually don't have one to show you. I wonder if I can just find one real fast. Yeah. Hey, Wikipedia is always good for things, right? Is it giving? Oh, yeah. Here we go. Okay, so can you see this right here fairly well? Okay, so again, what we have is we have an @ sign, your sequence ID, and then your genetic, what you actually read, that plus, and then just random characters that almost look like a regular expression search string, or someone swearing at you, or something like that. And these then just map to each one above was the quality of that. So here's the actual, let me see. Yeah, so here's the actual set of characters that encode quality. And you have everything from exclamation point, £, $signs, both parentheses, all of your alphanumeric characters, // backslashes, is greater than, is less than. Okay. So, interestingly, the reason that we don't put returns in the genetic data, which would kind of be nice to look at it, number one, you're generally not looking at. Number two, whoever defined the format did something a little bit unfortunate, is that it would be really nice if all the quality data was in characters that you wouldn't find in other rows. For instance, the @ symbol both denotes the beginning of your header sign and also your quality data. And then also we're using A, C, T, and Gs as both genetic data, but then also quality data. So it makes it a little bit more difficult to parse, not incredibly. But if you had an indeterminate number of lines, then it gets to be less fun.
Why do we need so much granularity with quality data? Why can't we just do like 0 through 9?
I haven't worked enough with high throughput data to know that it's super important that you have that much granularity. If you cut it in half, would it make it significantly worse? I don't know. And maybe when someone was defining the format, they didn't know either. They're like, well, it's better more than less. Maybe. I don't. Yeah. So long story short, for you listening, I just shrugged. There we go. Okay. And I already said this. Generally, they're used for high throughput data. We'll probably see these later on. Okay. The next one that's used very commonly. So FASTA and Nexus are probably the two most commonly used formats for sequence data. And these are going to be fairly significantly different. They're going to begin with a block name. So it begins with, generally actually it begins with #nexus, just to kind of let you know it's nexus, but that's actually not needed for the file format. That's just kind of convenient for humans to look at and be like, oh, this is nexus format. But the computers don't really care about that. The actual format begins with begin and then the name of the file, or the name of this block, actually, because you can have multiple blocks in one single file. And I'll show you in just a moment what that looks like. And then down below that, we have a line that has the dimensions of our data. So dimensions, it has the number of taxa. In the case of where I pulled this from, it had 4. And then the number of characters, or the number of bases per taxa, with this being representative of the maximum. If we have ones of different lengths, this is the maximum that you have for any one taxa. And then you have some format information as well. So the data type is, again, this can be DNA or protein or RNA as well. And we give it the data type here. We say, we define what our missing data is, and we define what a gap is. And we'll talk more about gaps later as we talk about alignment. And then you actually have a line that says matrix, and then below that you actually have your data, which is generally, you have a species name, and then the actual genetic code. After you get through all the different species, then you have just end semicolon, and that ends that block. So let me give you, is that the end of that? Oh, yeah, and then file name extensions for this, again, are all over the place. It could be .next, .nxs, .nexus, whatever people want. Okay, let's see here. Where's my Nexus file? I think it is... Wait, no. There we go. Okay, so this is a Nexus file. Again, we have #nexus, begin data. And in this one, we have dimensions, and we have number of taxa 14. And then the number of characters is 397. So the longest sequence in here is 397 bases long. Down below here, we have format, data equals DNA, and then gap equals a dash. And then we have matrix, and then we have each species, in this case, is now numbered. So 1, 2, 3, 4. And then after that, we have, and that number is in brackets, and then we have the header data, or the header data after that, and then the block of data, or the actual genetic data following that. So that is a Nexus format. Now, that being said, this can occur in two different ways. This is what's known as block format or sequential format. You can also have something called, let's see if I actually have this up. So again, that's blocked, or sequential is another thing that's called, where each taxa follows all of its genetic data. So you have the header information, then all the genetic data follows that. But you can also have what's called interleaved. And interleaved is each taxa then is only followed by some of its proportion of its data, whatever fits onto one line, usually I think again going up to 70 characters. And then you have the beginning of the next, and then the next below that, and so on. And then the next block again begins with all of those same taxa again. So let me look at, I think I have, actually I don't, hold on here. I think I pulled that up. Did I do a Nexus, copy one Nexus? Yeah, here we go. This is interleaved right here, where we have, again, nexus, begin data, we have our dimensions, we have our format, and then we have our matrix. And here, instead of having them the entirety of the data below that and then on, what we have is out beyond this, all of the data following this. Now, this being said, I'm going to show you a more common type of interleaving after this where, yeah, I'll come back to that. But essentially, we have everything on one line, and then oftentimes this will be repeated several times. But this is really nice because it allows you to look down and look at the alignment in the text file itself, which is not possible in block data. And we're going to come back to alignment in just a minute and talk about what that means or what that is. Okay. And nexus can either be interleaved or sequential, or interleaved in block. Okay. Okay, filet format is really, really simple. Filet format, your first line has just two numbers in, and that's it. The first number is the number of taxa, the next number is the number of bases. And this is only for genetic data, and then after that is generally interleaved. I'll show you what this looks like real fast. So, filet format looks like right there. Okay, so this is filet format. This is really simple. And again, this is interleaved, and this is more commonly what you see from interleaved data. So again, up here, first line, all it has is two numbers, 14,397. Really simple. It knows what it wants. And then after that, we have all of the header names, and then an area, and then some of the genetic data after that. So we have some block of that genetic data. But it's not the entirety of it. It's only out to one line, and then it returns and begins the next one. Now this, the genetic data for this one goes from here, and that picks up here. So you see there's another block down here. Again, this is the continuation of this first line up here, continues right here. And if you come down again, the continuation is right here again. Now again, the advantage of interleaved data is that it's easy in this text file to be able to look at the alignment, to actually look and see or compare between sequences, because homologous bases are lined up on top of each other, and you can look at fairly easily. However, that being said, if you want to scroll through or look at the entirety of one entire sequence, is really hard to do. If I want to pull one sequence out of here and put it into another file, incredibly hard to do. So we generally use this not very much anymore, because we have other things, other tools for looking at the alignment. So interleaved is not, we generally tend to use interleaved formats less and less. Okay, let's talk briefly about what you do with these, where you put them, where you get them if you're not sequencing them yourself, and that is databases. And most times when people go to publish their data, it requires them to make the genetic data publicly available. This is one of the first, genetics was really one of the first fields that made a very strong push that all of your data has to be, at least genetic data has to be completely open. More and more fields are doing this, more and more journals are doing this, but this has been kind of the default in genetic data since the beginning. You have to make it publicly available if you want to publish in most journals. And most modern publications are using genetic data, will almost always use at least some sequences that they did not sequence themselves. They've used data from a public repository, because we have so much sequence data out there. It doesn't, we are sequencing so much all of the time. It's really rare for some comparison sequence that you want to compare your sequences to is not publicly available. In fact, it's kind of weird when it isn't. We had a situation just recently. We were sequencing an octopus, and we wanted to match it up to some other sequences, and it was really weird that there wasn't those sequences out there. You have to look. Essentially, we had to find a deep sea octopus that no one has ever really worked on before we found something that no one had any sequences on. If people have even bothered to look at something for too long, they probably got sequences for it. It's just, yeah, it's so easy these days. And there's three major ones at the moment. We have GenBank. GenBank is, I'll talk more about that. This is the US one run by the NIH. There's EMBL, which is the European Molecular Biology Library. We also have DDBJ, which I think is the Digital Database for Biology of Japan. It's a Japanese one. But these tend to be the three most common. And so I'm going to talk a little bit more about GenBank specifically because it's one of the most prolific, it's one of the ones that most people go through to portal to everything else. Actually, all of these databases share information. So if something was originally archived in the EMBL, you can find it in GenBank. So it's kind of nice. And this is a database run by what's called the NCBI, which is National Center for Biotechnology Information, which is part of the NIH. Again, the NIH is running this whole thing, which, by the way, it's the National Institutes of Health. It's part of the U.S. federal government. And NCBI actually hosts a lot of useful tools for you to use. One of these is BLAST, which we will talk more about very briefly later on. And so let's look at GenBank. Let's explore a couple different things that are useful to use on GenBank. So there we go. You can get there just by Googling GenBank and the first result. So if you just look up GenBank, this is how I always get to it, is as the first result, looking up GenBank. And what you can do is you can actually look up nucleotide sequences, but also protein sequences. There's a little nice search thing here. You can look in all depositories, or you can look in specific different individual databases, sometimes for different data types, sometimes for different data species, and so on. But I just want to look in all the databases in the nucleotide. So imagine I wanted to find something, for instance, someone give me something that you would want to look up. Dictyostelium, okay. I don't want the S in there. And what gene would you want to look up? What's that? Which one?
RBRA.
Okay, so we look this up. Hey, I'm kind of curious. And then you just hit search and it's going to come up with, oh! Are you kidding me? It's RBRA, but it's not for Dicty.
You spelled Dicty wrong.
Oh, did I?
There's an O after the Y.
Dicty, oh! Is that right now? RBRA. Wow, why in the world? No one ever says it that way. Okay, so here we go. We have all of these nice sequences. And actually, I like we have a non-discoidium one here. That's cool. Fasciculatum. But we have RBRA for, and then we have other things that match not quite so closely. We can go ahead and get into here. Click on it, and then here's the format that they give you that gives you the locus. So most things that have their genome sequence, they actually have each locus on the genome has been given a number as well. Gives you the size, gives you the information that goes with whatever it is. The accession number, again, really important. And usually it's good to also include, so sometimes people can update updated versions of things, in which case it will be the accession number .1 is the first one, .2 is if they update, .3 is if they update a lot, .4 is if they're complete idiots. I'm just joking. I do not mean that. They are doing good things to keep things up to date. Good for them. I don't mean to. Oh, whoa. Hey, this was this one, the one here. Yeah, Ryan Lunsford was here when I was here. So was, yeah, Lisa McGill. Okay, Nate Whitney. Okay, so these are like people from my time. And then David. Cool. Makes sense because you gave me one that you're working with. So, makes sense. Okay, so you got this. You got another one. Is this his as well? That's a lot of people. So if you go into this, other things that are very useful about this is that if you come down, it gives you the reference information. So always do people publish when they initially find this, so it can be unpublished. But a lot of times what they'll do is they'll give you authors, they'll give the title and the journal and the reference information of the paper in which this initially appeared in. Yeah, and then it will give you, if it's cDNA, they will give you the translation, or often they'll just give you the translation, and there is your sequence. Go out of here, you can also get directly the FASTA file. And so what you see here is in FASTA format. And if you want to go grab, if you were wanting to grab, okay, so here's dictyostelium discoideum, we got 2 of those, and then we got fasciculum. If you select all these, okay, I got 3 dictyostelium RBRAs. Come down. At the very bottom of your page, you can go to the lower right-hand corner, send to. You can send to file. And then format, we can choose like FASTA. They have other ones in here as well. But let's choose FASTA. And then by create file. And we'll download it. And then we can open it. And it might tell me it doesn't know what to do. This computer probably doesn't know what it's doing. No, I just want Notepad. Come on. Ah! Windows. You make me weep. Open with, yeah. Here we go. Okay, that was useful. Please open with Notepad. Open with Notepad, or I'm going back to my old computer? Threats? Do threats work? Fine, whatever. You suck, Windows. Okay, so that didn't work. So that's how you obtain sequences of GenBank. And usually you get FASTA format or FASTA file out, and then you can start messing with it and doing cool things with it. So that's usually how you get things from there. Okay. So once you have sequences, there's a couple things that we can begin to do with these. And first off, there's some interesting things that you can know about the sequence itself. Kind of key parameters that are sometimes good to know about sequences is going to have a certain length or a certain number of base pairs or number of bases. And it's also going to have some certain base composition, which is also sometimes useful to know. You have a certain proportion of A's, percentage of T's, C's, and G's. So sometimes it's good to know that. And one other particular thing, kind of a subset of that, is oftentimes people want to know the GC content. Now the reason that people might want to know the GC content is that G and C bonds are stronger than AT bonds. And so what this means then is if something with more GC bonds are less likely to denature than things with a lot more AT bonds. And so in just practical application, this has application because this changes your denature temperature when you run something like PCR. If you have more GC bonds, it has to have a higher denature temperature. Less GC bonds, you don't need as high denature temperature. And then also this is important, like for instance, we have found some organisms that live in hot places or other things like this tend to have, or sometimes have, higher GC content that protects their DNA from denaturing. So sometimes that is important. But oftentimes what you want to do is not just look at characteristics of the individual gene. Yes?
How good are we at predicting like the actual denaturation temperature just from the sequence?
Pretty good.
Pretty good.
Yeah, there's actually a tool in Eugene that does that. Like, you give it it's actually, they have like these really kind of interesting PCR tools, and the reciprocal of this is that the anneal temperature is higher with more GC bonds, right? And so actually you can give it primers and it'll predict the anneal temperature that you need to do them at. And so like for instance, you have 2 primers, you want them to have similar anneal temperatures. You don't want the anneal temperatures to be like 10 degrees C off because that's going to cause problems. And so you can actually, as you're designing your primers, make sure they have similar anneal temperatures by using actually we're pretty good at predicting that. Now that being said, it's easier and easier for shorter stretches of DNA. As you get bigger and bigger stretches of DNA, it gets less and less because there's more things playing into the denaturation temperature than just GC content. So it's better the shorter stretches you have. The bigger it gets a little bit more stochastic because you have other things impacting it. So oftentimes what we want to do then, so there's other good things that we can find out about sequences just from one individual. Like for instance, the other really big thing is that we could find the reading frame and actually find out what protein would this code for. That's kind of nice, especially if you have cDNA, or what would be the amino acid sequence that you can code for, which that is a very relatively simple and straightforward process. But most of the other things that you're really interested in doing are going to involve some level of comparison of this genetic sequence to other genetic sequences. And this could be for a lot of different reasons. We want to know the relatedness between organisms or species or something else like this. And we're going to do that trying to look at the comparison between multiple genes from each of those. Also, we're really interested in adaptation. We want to look at the exact same gene in different populations of the same organism to see how are they different. How are they different? How might that be related to their biology or their environment, something else like this. Also looking at differences in functionality. Generally, when we look at functionality of a gene, it's in comparison to the functionality of something else. It's usually not just was its functionality in a vacuum. And again, to do that, to ever get its functionality, you need to be able to compare apples to apples and oranges to oranges. And so to do that, the first step in doing any sort of comparison between any genetic sequences is we need to do something called alignment. And alignment is so I have a big long thing definition here. Alignment is arranging sequences of DNA, RNA, or protein. So again, alignment happens whenever you have a molecular sequence. And we're doing this to identify regions of similarity that could be a consequence of similar functionality, structure, or evolutionary relationships between these sequences. So to boil that down to something a little bit simpler, all we want to do is we want to make sure that homologous either bases or amino acids in the sequence are paired. That we can say, okay, so this base right here, this A right here, is the equivalent to this T in this other sequence. For instance, you saw in the colloquium speaker last night for colloquium, comparisons between multiple genes of the same thing, looking at different amino acids that were in different portions of those receptor genes that she's talking about, and ones that are very conserved and ones that are not. Before you can ever make that comparison of what's conserved and what's not, you have to know where the alignment is. Where are analogous amino acids? Or in this case, analogous DNA base pairs. And that's coming back to one of the reasons that you might want to do this for functionality is really interesting to know. Even if you're looking at one you're only interested in one sequence and how that one sequence works. It's really important to know which bases are very conserved, therefore very important to the functioning of this molecule, this protein, and which ones are not conserved, which generally tends to give you ideas about which ones are not very important for the function. Or if they're not conserved and they have different functions, which ones might be leading to differences in function. And again, you need to be able to compare sequences to get that information. Okay. So, how does alignment work? And really, I'm not going to get too much down to the weeds of the math of alignment, but just give you the basics of this. And essentially what happens is that we're going to begin with whatever sequences you have. And the program was going to calculate or look at the similarities between sequences, and this is the alignment score. We're going to somehow calculate how are they similar. or in some cases, depending on how they're calculating, how are they dissimilar. That's the alignment score. And then the alignment program is going to introduce gaps to increase the similarity. It's going to start including gaps. Gaps are just null characters, just spaces, essentially, in the genetic sequence that allow for alignment that will increase the similarity between multiple sequences. Now, the key here is that the actual sequence is not changed at all. We are just essentially saying, where do we need a space in this so that it lines up better with another sequence? And the end result of what you get is some sort of array, where generally the rows are different sequences, and the columns are now homologous sets of positions. So let's kind of look at how this might look. So here we go. This is some stuff I just grabbed off of GenBank. This is some 12S system did talk. So this is ribosomal RNA. We're going to talk more about that soon. And it's just lines and stuff. And you know some of them are very similar. We have this GA that just is exactly the same all the way up and down. So really you're looking at where things are basically exactly the same up and down. Back in the day, we used to align things by hand. Not we, that's before my time. I don't know why I said we. I'm not that old. And you run this through an alignment program, and it gives you something like this. Okay, so this is aligned, and each dash you have here is now a gap. When you actually run this through any sort of other program that's looking at the sequence, it's going to ignore these gaps. But what it does is it spaces it out so that now, okay, so now look at this. We have this GA, and now this GA that didn't line up, it did line up in all these species, but didn't in this. Now we have an alignment right here that works really nice. We have the T, 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 GA, T, T, T. Same with T, 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 GA, T, T, T. And same all the way down. So now this aligns very well. And it's just done this by introducing these dashes or these gaps in here that we get this. And one thing that you notice is that there's insertions and deletions that are happening, and you don't really know which is an insertion or which is a deletion at this point. And so generally they're just called indels because we don't know if they're an insertion or a deletion. They're just an indel. What else was I going to say about that? Yeah. But again, none of the underlying sequences are changed, and now we can point out, okay, this one right here, this position, it is T all the way down. T is very conserved at whatever position this is. Whereas if we look at the next one, it's A, A, G, G. Okay, so this one's both A and G sometimes, and sometimes nothing. This is not a fairly highly conserved position. But to make those kinds of comparisons, we have to be able to align the sequence. I'm going to talk more specifically about how to do that in lab. And I'll pick up with the rest of alignment on Friday when we come in. So enjoy Wednesday. I will see you in lab.